Hello everyone, a pleasant morning, afternoon, evening, and or nighttime to you all. I am Miss Merle and this time we will be talking about cytokines with the hope that you will gain understanding how these chemical messengers orchestrate so many effects of our immune system and cells in it. The term cytokine is derived from a combination of two Greek words. We have cyto, meaning a cell, and kinos, meaning movement. So we have cyto and kinos. Cytokines are cell signaling molecules. They are chemical messengers that help or aid cell-to-cell communication in immune responses and stimulate the movement of cells towards sites of inflammation, infection, and trauma. So for the outline of my talk, we shall start with a definition of cytokines so that we will know what are they. Their attributes, families, production, gene expression, and followed by the secretion pathways of cytokines. And we will also go about what cytokine release syndrome is and get to know the pathophysiology of cytokine storm. Cytokines in the innate and adaptive immune responses, of course, innate and adaptive immune responses will also be discussed one by one. Moreover, cytokine production based on cell secretion will also be addressed. And relevant to include also are the cytokine and anti-cytokine therapies. And lastly, classical assays for cytokines will also be covered in a general aspect. So, what are cytokines? We've got to remember that an important effect of the interaction between microbes and tissue macrophages is the activation of macrophages and other immune cells to release small proteins called cytokines and chemokines and other chemical mediators. Collectively, these proteins they induce a state of inflammation. So think of inflammation in the tissue that attract monocytes and neutrophils to the infection and allow plasma proteins to enter the tissue from the blood. So well, that was quite a lengthy thing to remember, but going back with the definition of cytokines, they are small, they are soluble, and they are proteins secreted by specific cells. They can be proteins, peptides, or glycoproteins, and released by specific cells of the immune system. And they have a specific effect on the interactions and communications between cells. It's communicating, regulating, orchestrating. Those are the words for cytokines, and they are a category of, yes, since they are involved in communication, so they are signaling molecules that mediate and regulate. So immunity, inflammation, and of course hematopoiesis. So basically, cytokine is a general name, and other names are defined based on their presumed function, cell of secretion, and target of action. So if we are talking about cytokines with chemotactic activities, they are chemoattractants, so we call them as chemokines. And if we are talking about the cytokines produced by the lymphocytes, they are lymphokines, and those synthesized or made by the monocytes are monokines. We also have cytokines made by one leukocyte, and it will have an effect or it will act on other leukocyte or other WBC that will be an interleukin. Sadly, it's not an interleukine, right? So it has to be interleukin. So that's it for the definition of cytokines. So actions of cytokines. Cytokines can act in an autocrine manner, in an autocrine fashion, affecting the behavior of the cell that releases the cytokine. So autocrine action by binding to receptor on the same on the membrane of the same cell that secreted it. So that's for autocrine action. We also have paracrine action or paracrine manner. affecting adjacent cells or cells which are nearby or those cells in close proximity to each other. So that's what we call pericrine action. We also have some cytokines. They are even stable enough to act in an endocrine manner, affecting distant cells, so distant or see traveling through circulation, affecting on targets in distant parts of the body. So that's endocrine or endocrine activity. So cytokine actions include autocrine, pericrine, and endocrine manners. So next, let's go to the cytokine attributes. Cytokines are also endowed with distinct attributes or qualities or features regarded as characteristic or inherent in each cytokine. Cytokines can regulate cellular activity in a coordinated interactive way due to the following attributes or characteristics. Um, so how it is common for different cell types to secrete the same cytokine or for a single cytokine to act on several different cell types, or one cytokine has many different functions. That's what we call pleiotropy. So pleiotropy, first attribute or characteristic of a cytokine. Next, cytokines are redundant in their activity. They are redundant in their activity, meaning similar functions can be stimulated by different cytokines. So several different cytokines can mediate the same or similar function. So that's a redundancy. And they are often produced in a cascade, and one cytokine stimulates its target cells to make additional cytokines. And cytokines can also act synergistically and antagonistically. So synergism happens when there's a combined effect of two cytokines on cellular activity is greater than the additive effects of individual cytokines. And lastly, again, we have antagonism. The effects of one cytokine inhibits or offsets the effects of another cytokine. So again, for the cytokine attributes or characteristics or traits, we have pleiotropy, redundancy, synergism, and antagonism. We also have cytokine families. They belong to families. Large family of cytokines. In fact, they are these large families of cytokines are produced by various cells of the body, and the cytokine superfamily includes, of course, interleukins, chemokines or chemokines, and CSF or colony stimulating factors, interferons, transforming growth factors TNF, and TGF or tumor necrosis factor, and families. So, cytokines exist in broad families that are structurally related but may contain rather diverse cytokine functions. So they are related structurally, they have the same structure, may have similar structures, but when it comes to their function, they have diverse or different function. So that's the thing. Because families of cytokines share sequence similarity, again structurally related, and exhibit homology. So there's really a sameness in the structure and some promiscuity in their reciprocal receptor systems. But please remember, they do not exhibit functional similarity. So function, when it comes to function, they are not having the same. And cytokine families also contain important regulatory cell membrane receptor ligand pairs, reflecting evolutionary pressures that use common structural motifs in diverse immune functions in higher mammals. So that's for the cytokine families. Now let's have cytokine production, specifically on cytokine expression and gene expression. So cytokines, if you will ask me in what part of the cell are cytokines being synthesized or generated, they are synthesized in the Golgi, which are basically protein packaging organelles. and may traffic through the cytokines can be traffic, or they can go through the ER. From the Golgi, they can go through the ER, the rough in the smooth endoplasmic reticulum, to be released as soluble mediators, or they may remain membrane bound. So they can also remain in the cell membrane or plasma membrane, or they may be processed into cytosolic forms in the cytosol that can traffic intracellularly. Even they can return to the nucleus, where they can act as transcriptional regulators. Numerous factors promote cytokine expression in vivo. So what factors contribute or promote or advocate for the cytokine expression in vivo? So we have so many: cell-to-cell contact, autoantibodies, immune complexes. We have the local activation of the complement, the soluble products, and of course the microbial species producing them. We have the ROS, or reactive oxygen species, and their nitrogen intermediates. And take note, trauma and stress, and I know many medic students are suffering from stress, and also ischemia, radiation, UV light, ECM components, and the microbial and mammalian DNA, heat shock proteins, and of course cytokines themselves in autocrine loops. So those are the factors that promote expression of the cytokines in vivo. So now we have cytokine inducers. So what induces the secretion of cytokines? So induction in response to specific stimuli. They are induced in response to specific stimuli, such as bacterial lipopolysaccharides, or LPS, common in gram-negative bacteria. Flagellin, the component of the bacterial flagellum, and other bacterial products, and through the ligation of cell adhesion molecules, or through the recognition of foreign antigens by host lymphocytes. And please do note that bacterial LPSs, or lipopolysaccharides, also known as endotoxins, LPSs or endotoxins, are the major outer surface membrane components present again in almost all gram-negative, so gram-negative bacteria, and act as extremely strong modulators of innate or natural immunity in diverse eukaryotic species ranging from insects to humans. So that's it for cytokine inducers. And commonly, cytokines like IL-1, TNF, IL-6 are induced in the presence of LPS. We also have cytokine production. So what cells produce cytokines? We're done with the what part of the cell wherein cytokines are produced. Now let's go to knowing what cells produce cytokines. So predominantly, cytokines are made by many cell populations, but the predominant producers are helper T cells, CD4 positive, helper T cells, and the macrophages. So please don't forget. Again, predominant producers, manufacturers of cytokines, what cells, helper T cells, and the macrophages. Cytokines may be produced in and by peripheral nerve tissue. So that's something we have just known, in and by peripheral nerve tissue, during normal and pathological processes, by resident and recruited macrophages, mast cells, endothelial cells, and Schwann cells. Following a peripheral nerve injury, for example, macrophages and Schwann cells that gather around the injured site of the nerve secrete cytokines and specific growth factors required for nerve regeneration. So it's very beneficial. And localized inflammatory irritation of the dorsal root ganglion not only increases pro-inflammatory cytokines, but also decreases anti-inflammatory cytokines. So FYI. And now let's go to the secretion pathways of cytokines, how they're being secreted. So we just like the complement with the classical and the non-classical. Somehow they are the same, but still different. So recently, more information has come to light from innate immune cells regarding the distal steps of cytokine secretion from the Golgi complex through membrane-bound organelles for classical secretion involving membrane fusion and exocytosis. So here, cytokines may be packaged in the Golgi for storage in secretory vesicles or granules and then secreted only during receptor-mediated release in a form of regulated exocytosis, so from the Golgi. Or they may be released rapidly, so a very rapid release upon their synthesis through recycling endosomes and small secretory vesicles through constitutive exocytosis. So we have regulated and constitutive exocytosis if the secretion is done in a very rapid manner. And next, cytokines may also be released through alternative pathways, not following the classical pathway, of course, and released through alternative pathways such as molecular transporters in a non-classical secretion. So that's for the cytokine secretion. So now we have next. Cytokine release syndrome. What is it? It is a syndrome which is caused by a large, rapid release of cytokines. But remember it has to be rapid and large. From cells targeted to the antibody as well as immune effector cells recruited to the area. So again, cytokine release syndrome is caused by a large, rapid release of cytokines into the blood from immune cells affected by immunotherapy, a condition that may occur after treatment with some types of immunotherapy, such as monoclonal antibodies. If monoclonal antibodies are used, see, it is a symptom complex associated with the use of many monoclonal antibodies and or CAR T cell therapy. So, signs and symptoms include, of course, febrile reactions from the patient, fever, there's nausea, there is hypotension, chills, tachycardia, asthenia, headache, scratchy throat, and dyspnea. However, you shouldn't have to be complacent with cytokine release syndrome because this one is an oncologic emergency. It's a life-threatening reaction. Whenever there is the presence of the positive, massive cytokine release, it's not good. It can lead to death. It is fatal. And you might ask me what type of instances or scenarios in the hospital setting that this one might happen. So it is common during the first infusion in patients, their primary infusion in patients with hematologic malignancies who have not received prior chemotherapy. So always look out for cytokine release syndrome for these kinds of patients. Next, we have the cytokine storm or hypercytokinemia. So the cytokine storm, or again hypercytokinemia, is a systemic expression of a healthy. Take note here, the immune system is healthy and vigorous. However, it resulted to the release of more than 150 known inflammatory mediators. This is a party that can lead to death. Why? Because what type of inflammatory mediators? We have cytokines, oxygen-free radicals, and coagulation factors. We have both pro-inflammatory cytokines. The pro-inflammatory, take note, the TNF-alpha, IL-1, IL-6, and we also have anti-inflammatory cytokines such as interleukin 10 and 1 receptor antagonist. And all these pro-inflammatory cytokines and anti-inflammatory cytokines are elevated in the serum of patients experiencing a cytokine storm. Isn't it contradictory? Like pro-inflammatory and anti-inflammatory all together. And this overproduction of cytokines causes positive feedback on other immune cells. This positive feedback, which permits for more immune cells or immune mediators to be recruited to the site of injury that can lead to organ damage. Because take note that excessive production of pro-inflammatory cytokines leads to ARDS aggravation and widespread tissue damage, resulting in multi-organ failure. And eventually it will lead to death. Targeting cytokines, that's why right now it's very relevant that now we are affected by this pandemic, and we have SARS-CoV-2 infections going uncontrolled. So here, targeting cytokines during the management of COVID-19 patients could improve survival rates and highly reduce mortality. Cytokine storms are just one of the examples that when aberrant or exaggerated response of the host immune system, it can cause severe disease if it remains uncontrolled. So always, things will always fall into place if they are properly controlled. But once things have become crazy, so that's the thing, like cytokine storms can happen. Next, what are the symptoms? So cytokine storm syndromes, they are a group of disorders representing a variety of inflammatory causes. The primary symptoms of a cytokine storm are hyperpyrexia or high fever. There is swelling, redness, extreme fatigue, and nausea. In some cases, the immune reaction may be fatal. So again, fatal, hyperpyrexia, and we can also look for the markers of a cytokine storm, the presence of the following: ferritin, D dimers, LDH, CRP, cytokines themselves, ESR elevations, neutrophil and lymphocyte ratios, and ALTs, which you will be learning in your clinical chemistry, and some from IS and hematology. And what are the causes of cytokine storm? Well, well, when the immune system is fighting pathogens, cytokines signal immune cells such as T cells and macrophages to travel to the site of infection. In addition, cytokines activate those cells, stimulating them to produce or make more cytokines. Normally, this feedback loop is kept in check by the body. However, in some instances, the reaction becomes uncontrolled, becomes highly uncontrolled, and too many immune cells are activated in a single place. Too many immune cells, too many cytokines produced in a single setting. So, is this good? No. The precise reason for this is not entirely understood, but may be caused by an exaggerated response. So the response became exaggerated when the immune system encounters a new and highly pathogenic invader. And cytokine storms have potential to do significant damage again and again, as what I have told you in the previous slides. It can lead or cause significant damage to body tissues and organs. If a cytokine storm occurs or happens in the lungs, for example, fluids and immune cells such as macrophages may accumulate and eventually block off the airways, potentially resulting to death. So it's not really good. And cytokines in the now let's go let's discuss the cytokines in the innate immune response first, and followed by the cytokines in the adaptive immune response. So cytokines involved in the innate immune responses or response are responsible for many physical symptoms attributed to inflammation. Like, remember, we have inflammation, fever, we have swelling, we have pain, and the presence of cellular infiltrates into damaged tissues. The innate immune response is a non-specific response, but it happens within hours or the first contact with the microorganisms. And so what cytokines are involved here in triggering the innate immune response surely are the following: IL-1, IL-6, TNF alpha, the chemokines, the TGF factor, beta, in interferons alpha and beta. So let's begin with IL-1. So IL-1 family consists of IL-1. We have three, subunits or isoforms. And these cytokines are produced by the IL-1 alpha and IL-1 beta. They are produced by monocytes and macrophages. By the way, this is the abbreviation for macrophages. It's like this, macrophages. And then IL-1 alpha, it remains intracellular within the monocytes normally. And whenever there is a programmed cell death or apoptosis, the IL-1A can be released, and the release will lead to the attraction of inflammatory cells to the areas where infection or inflammation or injury is ongoing. And then remember that IL-1 in general is responsible for most of the systemic activity attributed to IL-1, including fever, activation of phagocytes, and production of acute phase proteins. And it is cleaved intracellularly to an active form that is then secreted by monocytes. And this one is very important. IL-1 acts as an endogenous pyrogen. So it induces fever, high body temperatures in the acute phase response through its action on the hypothalamus. The hypothalamus is the body's thermostat, right? Thermostat. And then for the IL-1 sets the thermostat at a higher level. So blame IL-1 why you are having fever. Elevated body temperatures, what's the benefit, or is it useful? Because it serves to temperatures serve to inhibit the growth of pathogenic viruses in bacteria and microbes. And along with that, it also increases lymphocyte activity. Additionally, IL-1 induces the production of vascular cell adhesion molecules or VCAM, as well as chemokines and IL-6. These chemokines and cell adhesion molecules attract and assist leukocytes to the area to enter the inflamed areas by a process known as diapedesis. So, changing the thermostat, IL-1, responsible for diapedesis, IL-1, an endogenous pyrogen, it's IL-1. So that's it for interleukin-1. We also have the tumor necrosis factors, or TNF. They were first isolated from tumor cells, and were so named because they induce a rupture or lysis of these cells. And tumor TNF alpha is the most prominent member of the TNF superfamily. And always remember, if we are talking about vasodilation, and increased vasopermeability, think of TNF alpha. And then this TNF alpha can exist as a membrane-bound form or in soluble form. So we have two forms of TNF alpha. By the way, before going to the forms, the main trigger for TNF alpha production is the presence of, again, very notorious, the LPS of the gram-negative bacteria. TNF alpha secreted by activated monocytes and macrophages can activate T cells through its ability to induce expression of MHC class II molecules. So that's it. And then going back to the forms, we have the membrane-bound and the soluble forms. Membrane-bound, what's the effect? It can mediate all the cytotoxic and inflammatory effects of TNF through cell-to-cell contact. And however, the soluble form is not really that useful because it is unstable and it has a short half-life. So think of the membrane-bound TNF alpha doing all these things and forget the soluble forms. And And as what I have said, it can activate T cells, able to induce expression of MHC class 2 molecules. So what's the thing with this? It can lead to an increase or too much TNF. This condition can lead to very large amounts of TNF secreted in response to gram-negative bacterial infections, leading to or resulting to a decrease in blood pressure. There is hypotensive reduction in tissue perfusion and disseminated intravascular coagulation. So the latter, or DIC, can lead or result to uncontrolled bleeding. So what's the main point here I would want to let you understand is that if there is this large amounts of TNF secreted, it can lead to septic shock. Septic shock is deadly. Next, we also have TNF alpha, and Stevens said that this is the central mediator of pathological processes in rheumatoid arthritis and in Crohn's disease. So, TNF alpha and IL-1 together with IL-1, TNF alpha plus IL-1, they are both present in rheumatoid synovial fluids. So, in the synovial fluids and synovial membranes of patients with rheumatoid arthritis, IL-1 and TNF-alpha are present, and studies with anti-TNF-alpha and anti-IL-1 demonstrate or show that TNF-alpha is the central mediator of pathological processes in RA and Crohn's disease. So blame why there is inflammation and all those things going on with those patients having RA, suffering from RA and Crohn's disease. It's TNF-alpha. Please remember that rheumatoid arthritis is a chronic, progressive, and disabling autoimmune disease. It causes inflammation, swelling, and pain in the joints and can really affect other body organs, while Crohn's disease is a type of inflammatory bowel disease, or IBD. Inflammatory, RA, inflammatory, Crohn's disease, inflammatory disease. Since Crohn's disease is a type of IBD and it causes inflammation of your digestive tract, which can lead to abdominal pain, severe spells of diarrhea, fatigue, weight loss, and malnutrition. So let's go to IL-6. IL-6 is a single protein produced by both, take note, both lymphoid and non-lymphoid cells. It is a part of the cytokine cascade released in response to LPS. So LPS again, IL-1, IL-6, and TNF, LPS. Please remember. Again, and it plays an important role in acute phase reactions in the adaptive immune response. So acute phase reactions. And then it is expressed by a variety of normal and transformed cells. So normal cells express IL-6, transformed cells express also IL-6, like T-B cells, monocytes, macrophages, hepatocytes, keratinocytes, astrocytes, vascular endothelial cells, and various tumor cells. So these the tumor cells are the transformed cells, normal cells are the following. And then what's the main thing here? Because in the presence of IL-1, IL-6 is being triggered, so it's being secreted. Again, IL-1 primarily triggers its secretion. And then this pleiotropic cytokine affects inflammation, acute phase reactions, synthesis of immunoglobulins or antibodies, activation states of B and T cells. And please remember what is pleiotropy or pleiotropic. What do you mean by a pleiotropic cytokine? Pleiotropic cytokine, it is a cytokine that affects the activity of multiple cell types. It has many effects. And also please remember IL-6 stimulates B cells to proliferate into plasma cells. So somehow CD4 helps the B cells to mature. IL-6 also stimulates B cells to proliferate and differentiate into plasma cells because IL-6 induces CD4 T cells to produce greater quantities of both pro and anti-inflammatory cytokines. So that's it for IL-6. Next, we have the chemokines. They are a family of cytokines that the thing is they enhance, they accentuate, they facilitate motility and migration of many types of leukocytes or WBCs toward the source of the chemokine. So they facilitate chemotaxis. That's it. They facilitate chemotaxis. Most of the chemotactic activity is regulated by the activities of chemokines, including the response to infectious diseases, autoimmune inflammation, CA or cancer, and the homing of lymphocytes to all the lymphoid tissues. So, chemokines, they are classified into families. So we have four families: the first, second, third, and the fourth. The first family, also known as the CXC alpha chemokines. Why CXC? Always remember, C stands for cysteine, and if we are talking about alpha chemokines or chemokines, a single amino acid, there is the presence of a single amino acid between the first and the second cysteine. So here, and amino acids are represented by letter X. So that's why it's CXC. We also have beta chemokines, so CC. Why CC? Because nearby or sitting beside each other, adjacent cysteine residue. So one, the first and the second, they are together, so adjacent. So that's what we call the CC or beta chemokines. We also have the C chemokine family. So C, because it lacks one of the cysteine. So it only has the first cysteine, but it doesn't have the second cysteine. We also have the lastly the CX3C chemokines, meaning in between the first and the second cysteines, we can see or observe three amino acids. So that's why it's X, because X equals an amino acid, and we have three amino acids there in between C and C. So that's for CX3C chemokines. And then the importance lies in HIV, which I will be talking about later about chemokines. So chemokines, they permit the extravasation of leukocytes or WBCs into the tissues. So remember that what is a leukocyte extravasation? It is also commonly known as leukocyte adhesion cascade or diapedesis. It is the passage of cells through intact vessel walls, and it is the movement of leukocytes out of the circulatory system, out from the blood vessels, and towards the site of tissue damage or infection. Here, leukocytes rolling on capillary endothelial cells activate their chemokine receptors in the presence of chemokines. This, in turn, activates integrins or cell adhesion molecules on leukocytes and leads to firm adhesion on endothelial cells. So the endothelial cells here. And then, shared expression of chemokine receptors among different types of leukocytes permits or allows for the co-localization of multiple cell types to the damaged tissue and helps to broaden the response to tissue damage. So that here, this is the good thing. Here is the bad thing. Why? This one is physiologic in nature. This one represents something pathologic because here, the chemokine receptors CXCR4 and CCR5 are utilized by HIV. So the HIV virus uses these receptors as co-receptors for infection of the CD4 T lymphocyte. So the cell is your CD4 lymphocyte. and macrophages. So people or patients with certain polymorphisms in these chemokine receptors, CXCR4 and CCR5, are long-term non-progressors, so meaning they will not show signs and symptoms. They will remain asymptomatic and have normal CD4 T cell counts and normal immune function and have low or undetectable viral loads. So somehow it's good. And then the altered protein sequences of the receptors, if they are, take note, it's good if they are not, or if they are having polymorphisms. But in the event that they are not having polymorphisms, then surely it's easier for the virus to enter and infect and to start the viral replication in those cells, because these chemokine receptors are utilized by the virus. The CXCR4 and CCR5 receptors are used by HIV to easily gain entry into the cells. And then remember that those patients who are having polymorphisms or not having these receptors, their CD4 T cell counts and normal immune function, they have normal CD4 T cell counts, and also their immune functions are normal and have, again, I'm just saying this one again and again, and have low or undetectable viral loads. So they have this increased chance of survival. So meaning, if you are having a polymorphism, it's okay, pero if not, then surely you will die. Not die, but you will die slowly with AIDS. Next, we have the TGF-beta, transforming growth factor beta. Chemokines, how to say, these are still cytokines in the innate immune response. And for the TGF-beta, we have three types, the three isoforms: TGF-beta 1, 2, and 3. But the one which is much celebrated and highly studied is your TGF-beta, because it induces a growth arrest in tumor cells, so killing, or those those tumor cells will not be able to multiply and grow. It induces anti-proliferative activity in a wide variety of cell types. So anti-proliferative activity in a wide variety of cell types. Whenever TGF-beta is activated, it primarily regulates these things: cell growth and differentiation, programmed cell death or apoptosis, migration of the leukocytes, and other inflammatory mediators, and as a whole it functions in the inflammatory response. Thus, we can, how to say, conclude that it functions as a control to aid down regulate the inflammatory response. If the inflammatory response is getting too high, it is the TGF-beta that will bring back homeostasis. It will try to down regulate the inflammatory response whenever, especially if it's no longer needed. So TGF-beta functions as both an activator and an inhibitor of proliferation. Somehow contradicting again, but well, that's life. That's life for the TGF-beta cytokine. So activator functions of these cytokine include the expression of CD8 in CD4 negative, CD8 negative thymocytes. So why we are having CD8? It's because of TGF-beta. Inhibitory functions or inhibitor functions of this cytokine TGF-beta include also inhibitory. It functions as an autocrine inhibitory factor for immature or those baby thymocytes. Macrophage activation, also somatic cell growth, IL-12 production, and inducing interferon gamma. So next, we have the interferons. So interferons were originally so named because they interfere with viral replication. So viral replication, how viruses replicate inside the cells will be blocked. So however, it is the type 1 interferons consisting of interferon alpha and beta that function primarily in this manner. And then these cytokines are produced by dendritic cells or DCs. And generally, interferons create a condition of resistance to viral infections in cells by activating endonucleases and suppressing the translation of viral RNA and restricting viral entry into the cytosol. So type 1 interferons, again, they are produced by dendritic cells, and they induce production of proteins in pathways that directly interfere with viral replication and cell division. And in most cases, this helps limit the infection to one relatively small area of the body. Type 1 interferon activates NK cells and also enhances the expression of MHC class 1 proteins, thus increasing the recognition and killing of virus-infected cells. So much for the interferons. No, still, we're not yet done with the interferons, sorry. Type 1 interferons, they function to activate NK cells. They enhance the expression of MHC class 1 protein, and they are also active against cancers. Studies have shown that interferon beta is effective against it is efficacious in treating multiple sclerosis, although the exact mechanism of action remains unclear. And while interferon alpha has been used to treat hepatitis C and Kaposi sarcoma, as well as certain leukemias and lymphomas. So, cytokines involved in the innate immune response are produced by many different cell types and function mainly to increase acute phase reactants and to recruit leukocytes or WBCs to the area of infection. And in contrast, cytokines or cytokines in the adaptive immune response, their main function is they directly affect T and B lymphocytes. So yeah, to paraphrase it, cytokines involved in the adaptive immune response are mainly secreted by T cells, especially T helper cells, and affect T and B cell function or lymphocyte function more directly. So here for adaptive, but here it's more on acute phase proteins, acute phase reactants, and the recruitment of those WBCs or other inflammatory mediators to the site of injury or infection or trauma. So we have the three main T helper cell subclasses. Because I know that you're just familiar with the T lymphocytes. We have the CD4 and the CD8, right? And CD4 is your helper. CD8 is your cytotoxic, but your helper still has these three main cellular subsets or subclasses. So again, there are three main subclasses of the T helper cells: Th1, Th2, and we also have the Treg or T regulatory cells. Each has a specific function and produces a different set of cytokines. So they don't function in the same manner, and also they produce different or a variation of cytokines. Once the T cell receptor captures the antigen, clonal expansion of those particular CD4 T helper cells happens. And then differentiation into Th1, Th2, or Treg cell lineages is influenced by the spectrum of cytokines expressed in the initial response. So it's based on what kinds or what types of cytokines are expressed during the initial response. And then the Th1 lineage is driven by the expression of IL-12 by disease and is primarily responsible for cell-mediated immunity. While Th2, so this one cell-mediated immunity, while Th2 drives antibody-mediated immunity. So here antibody-mediated, that's why we have the IgE here. And what else? And Treg cells are derived from IL-10 responsive naive T cells and help to regulate the activities of Th1 and Th2 cells. So here is what we can see in this table from the book of Abbas. TH cell signature cytokines. So mainly these are the signature cytokines associated per TH cell subset or subclasses, and they have different immune functions because this one IgG production, this one is for IgE, this one is more on inflammation, and then host defense for intra, so you should have to be really familiarized with this one for intracellular microbes. TH1 extracellular, specifically fungi and bacteria, we need TH17. So role in diseases, so the following. We also have next the interferon gamma. So activation of TH1 induces high level expression of interferon gamma. And interferon gamma is the principal molecule produced by TH1 cells. Let's go back. Interferon gamma, see the signature cytokine of your TH1 cell. So again, interferon gamma is the principal molecule produced by TH1 cells, and it affects the RNA expression levels of more than 200 genes. Take note. Interferon gamma stimulates antigen presentation by MHC 1 and 2 molecules, and these increased expression of the MHC class 1 and 2 molecules on antigen presenting cells increases the likelihood of antigen capture and the involvement of additional lymphocytes. So what are the two ways of stimulating interferon gamma production in mature, take note, mature TH1 T helper 1 cells is through a ligation process, ligation of the T cell receptor by MHC peptide antigen presentation. We also have stimulation of the following cytokines so that they can act synergistically to stimulate interferon gamma production. We also have another TH1 cytokine, IL-2, under the cytokines of the adaptive immune system. TH1 cells also secrete IL-2, yeah, in addition to interferon gamma. IL-2 is also known as this is very important, a board exam question. What is IL-2? IL-2 is also known as the T cell growth factor since it drives the growth and differentiation of both, take note, both T and B lymphocytes. Different growth and differentiation, take note, and it induces rupture or lytic activity of natural killer cells. IL-2 alone can activate proliferation of T helper 2 cells and helps to make IgG1 and IgE producing cells. The clonal expansion of activated T helper cells is a necessary portion or part of mounting an adequate immune response to any immunologic challenge. So it has to be thought that that's the way IL-2 can activate the proliferation of Th2 cells. And next we have IL-4, still a Th2 cytokine. So as mentioned previously, Th2 cells are primarily responsible for antibody-mediated immunity, right? And IL-4 is one of the key cytokines regulating Th2 immune activities and help drive antibody responses in a variety of diseases. IL-4 promotes the production of IgG2A, so IgG2A and IgE, and along with IL-5, it drives eosinophilic, so that eosinophils can differentiate and be activated in both allergic immune responses and also as a response to parasites. So think of eosinophils, parasites, and isotype switching, all attributed to because of the presence of IL-4. These are related: parasites, IgE, allergy, IgE, so isotype switching. So IL-13 is how to say, somehow there is a similar cytokine to IL-4, and that's IL-13. IL-13. Is a cytokine with many of the same properties as IL-4, and both of them, IL-4 and IL-13, they induce worm expulsion again, so that these worms can get out from the body. Worm or parasite expulsion, and they really on the side on they facilitate IgE class switching. By the way, what is isotype switching? So isotype switching is the process whereby an activated B cell changes its antibody production. Normally it's from really IgM. It starts with IgM. So here again, isotype switching is the process whereby an activated B cell changes its antibody production from IgM to either IgA, IgE, IgG, depending on the functional requirements. And so now we have IL-10. IL-10 somehow is if we will summarize what is IL-10, it is the a down regulator of the immune response. So it has anti-inflammatory and suppressive effects on Th1 cells. Really? Yes. It also has, it can inhibit antigen presentation, and it can stimulate CD8 positive T cells. It can produce B cells, B cell MHC2 production, but the major effect is really on this one. Since it contradicts, it antagonizes, it is an antagonist to interferon gamma. The promotion of a Th2 cytokine pattern leads to the thought that IL-10 is a downregulator of the immune responses. So again, IL-10 downregulates the immune. This is the only known cytokine that downregulates the immune response. That's IL-10, according to Stevens. So here we have the cytokines associated with Tregs or T regulatory cells. So what are these Tregs? Tregs, they are the third major subclass of CD4 T cells. Again, Tregs or T regulatory cells. Tregs are CD4 positive, CD25 positive, CD4 positive, CD25 positive T cells. So these two surface markers are present in the cells. Okay, we have this, for example, CD4, and this one is your CD25. So Tregs are, again, they are selected in the thymus, so thymic selection. And they play a key role in establishing peripheral tolerance. So peripheral tolerance is brought about by the functions of regulatory T cells. To peripheral tolerance to what? So to a wide variety of antigens, allergens, tumor antigens, transplant antigens, and infectious agents. CD4 positive, CD25 positive Tregs affects T cell activity primarily through the actions of TGF-beta. So TGF-beta induces expression of FOXP3. What is FOXP3? It is a transcription factor that causes Treg cells to suppress the activity of other T cells. Tregs may be found in transplanted tissues and help to establish tolerance. So whenever you can see T tolerance. So that's one. Again, Tregs in transplanted tissues, we can find them, and what is their business there? Tregs help to establish tolerance to the graft by the host immune system through the alteration of antigen presentation. So that's it for the Tregs and the TGF-beta. Next, we have the EPO or erythropoietin, and we also have the colony-stimulating factors. So the colony-stimulating factors, CSF, include IL-3, EPO, and granulocyte macrophage and granulocyte macrophage colony-stimulating factors, abbreviated as GCSF, MCSF, and GMCSF. And GMCSF, or the granulocyte macrophage colony stimulating factor, acts to drive differentiation toward other white blood cell types. So if here Take note, activation, and it becomes, so if your MCSF is activated, it becomes these, it will lead to the development or formation of macrophages. If your GCSF is activated, GCSF will become neutrophils. GMCSF plus IL-3, they will become or transformed into basophils and mast cells. GMCSF plus IL-5 to IL-3, they will become eosinophils. So that's for the colony stimulating factors. And EPO, what's the role of EPO? Well, EPO is produced by what organ? Well, EPO regulates RBC production in the bone marrow, but is produced primarily in the kidneys. So what produces EPO or erythropoietin? The kidneys, but it regulates RBC production in the red bone marrow. EPO alpha is the only one which is FDA licensed and often prescribed to improve RBC counts or erythrocyte counts for patients with anemia. So a low RBC concentration, and they will be given EPO or erythropoietin alpha, and for those with cancer who have undergone radiation and chemotherapy. So it's very important because what's the rationale of giving EPO to anemic patients? So RBC proliferation induced by EPO improves oxygenation of the tissues and eventually switches off EPO production. The normal serum EPO values range from 5 to 8 units per liter, but if you're a physician, you must interpret this in relation to the hematocrit, as levels can really increase by up to 1,000-fold during anemia. Next, we also have, well, the main thing why I presented this one is for you to remember more on mnemonics, so that you will know that what are the cytokines produced or secreted by macrophages, 1, 6, 8, 12, and the TNF alpha. So this kind of presentation is somehow showing you it's arranging the cytokines based on the cells that secreted them. So what are the important cytokines to remember here? So 1, 6, and then we have IL-1, IL-6, TNF alpha, then recruit IL-8 and IL-12. So we have secreted by macrophages. Again, macrophages here. Macrophages, again, what's the symbol? Macrophages. And then here, for you to remember, hot T-bone steak. Why? IL-1, fever, so it's hot. IL-2 stimulates T cells. IL-3 stimulates bone marrow. IL-4 stimulates IgE production, while 5 is IgA production stimulation, and IL-6 stimulates acute, so that it will become C becomes K. So hot T-bone steak. You'll never forget that. 1, 2, 3, 4, 5, 6. See? An easy way to remember, because honestly, I myself before, I cannot really memorize all these cytokines. I even thought chemokines and cytokines are the same when I was still in third year, way back in third year. And now you can also observe that IL-1 is implicated in fever, IL-6 fever, TNF also is, there's also an implication in fever, but it's not shown here. And then also for interleukin 8, you can remember clean up on IL-8. Neutrophils are recruited by interleukin 8 to clear infections. So that's a way for you to remember that. Again, however, for fever, basically we have 1, 6, and TNF. For fever, we have 1, 6, and TNF alpha. And next, we have those cytokines secreted by all T cells. So there's no mnemonic for this one. So we've just got to go over. IL-2 stimulates growth of helper cytotoxic and regulatory T cells, because this is the T cell growth factor. IL-2, very important. IL-2, the T cell growth factor. And then IL-3 supports growth and differentiation of bone marrow stem cells and functions like GM-CSF. And now our interferon gamma is secreted by NK and T cells and then activates interferon C, viral replication blockage, and activates NK cells to kill virus-infected cells. And lastly, we have the cytokines from T helper 2 cells, so IL-4, 5, and 10. So for you to remember, for IL-4, and too proud to beg for help, and too proud to beg for help. Why? Helper 2 cells promotes growth of B cells and enhances class switching to IgE, and IgG. So this one. So it's easier to be remembered that way. We also have IL-5, promotes differentiation, growth and differentiation of B cells, and enhances class switching to IgA. So from IgM, it will become IgA. And then for IL-10, this one is the downregulator of the immune responses, remember? So TGF-beta and IL-10 both attenuate the immune response. See? TGF-beta and IL-10B both attenuate the immune response. So that's it. We also have cytokines and anti-cytokine therapies. Well, I will not go over with the specifics of all these kinds of therapies, but it's really good to know that cytokines are used in immunotherapy, and nowadays immunotherapy is really an edge on pharmacology. So we here in the Philippines cannot really see the significance, but in modern and affluent countries, they are using immunotherapy in a very high level in treating autoimmune diseases and most cancers. So let's go back to the main thing. Recombinant DNA techniques have allowed for the production of humanized monoclonal antibodies that are much less immunogenic and that function as cytokine antagonists. So an example is infliximab or Remicade. This one is a chimeric antibody. We will be, I hope, touch chimeric antibodies. Chimeric antibody containing human constant regions, and murine meaning from mouse or mouse-derived antigen-specific arms that bind human TNF-alpha. So what's the mechanism of action? For a while, my dog. So for the mechanism of action, infliximab, it's easy for you to remember. Mab, monoclonal antibody. Anything that ends with mab, always remember monoclonal antibodies. That's why this one is a chimeric antibody containing human constant regions and murine antigen-specific arms that bind to human TNF-alpha. So here, how does this drug work? So this one, it blocks the activity of TNF-alpha in RA and Crohn's disease. So a single dose alleviates symptoms and reduces swollen joints. Very effective, but very expensive. Take note, monoclonal antibody generation through hybridoma technology, a very expensive thing to do. And then we also have Xenapex. Here, monoclonal antibodies can be further humanized so that the complementarity determining regions are the only murine components remaining. So much humanized portions, less on the murine or the mouse. So murine mouse, right? And then an example of that is Xenapex, which is directed at human IL-2 receptor. IL-2R is for the receptor. These antibodies generally have a lower affinity and extended half-life, but reduce immunogenicity, so that's the advantage. And another approach to anti-cytokine therapy is the development of hybrid proteins with cytokine receptors. Binding sites or receptor binding sites attached to immunoglobulin constant regions. And what's the composition? It consists of the extracellular domains of type 2 TNF receptor, type 2 TNF receptor fused to the heavy chain constant region of IgG1. These things might be difficult for you, but I know it will make sense later as we go over with the other discussions. And the MOA, or the mechanism of action, well, this one, etanercept, or NREL, is a fusion protein, and it can bind to TNF alpha. TNF alpha, similar to infliximab. TNF alpha, and block its activity. But guys, remember, it's easy to say all these things, but the generation itself of the monoclonal antibodies, how you will grow them, how you, no, not grow, but the cells have to be grown, the myeloma cells, and then there's the fusion. I might be the one to discuss that, but I'm not sure if it's me or Ma'am Pam or Sir Vincent. And then let's go to the classical assays for cytokines. So we have basically these assays are fixed to research labs and not on more on the clinical setting. We cannot see this in Sacred Heart, in Vicente Soto, in VCMC, even in Chonghua, I think, or CBDOC. So it's more on research labs the research labs are doing this, but not really in the Philippines. And so, although there are several cytokine assay formats available in Europe, none have been approved for clinical use by the FDA at this time. See, that's why it's in the research labs. And what is the most basic test is the ELISpot assay. It uses the enzyme-linked immunosorbent assay technique. So ELISA is the principle on in vitro activated peripheral WBCs. And recall that, what is ELISA? ELISA, sorry, I think you haven't had the discussion for this. So we will know ELISA later in our discussions. So ELISA is short for enzyme-linked immunosorbent assay. It is a plate. You have the microtiter plates here. It is a plate-based assay technique designed for detecting and quantifying soluble substances such as proteins, antibodies, hormones, and peptides. So for example, this one is one well in a bigger, is just one of the wells from this microtiter plate. So it's not as big as like it's a beaker or something. No, reactions happen in the microtiter plates. And these the microtiter plates are having individual wells. Basically this one is a 96 well microtiter plate. And then I'm always doing that. It's so bad because I cannot show you the things I've done in Mahidol in Thailand in the research lab that I am working. But hopefully if you'll become interns, and I will be showing you the real stuff for this. But now we will not discuss this that much because we are just talking about the assays for cytokines and not the specifics about the ELISA or ELISpot assay. So going back, this one. So what's the procedure? In the ELISpot process, either a monoclonal or a polyclonal, so we can use any monoclonal or polyclonal antibody specific to the chosen cytokine is pre-coated onto a microplate. So what is in the microplate? It could be your monoclonal or polyclonal antibody specific for the chosen cytokine is pre-coated into a microplate. So it's in here. And throughout the period of incubation, the immobilized antibody, in the immediate vicinity of the secreting cells binds the secreted cytokine. And after washing step to remove the unbound antibodies, we have a colored precipitate formation and appears as spots at the sites of the cytokine localization. So if we will have a top view, spots, in the spots, each spot will represent an individual cytokine secreting cell. So the spot formation, the endpoint will be the spot formation, and it heralds a cytokine secretion. So that's it for your ELISpot assay. Next, we have the multiplexed ELISA. Well, these ELISAs utilize or use several detector antibodies. So now we have many antibodies here. Detector antibodies bound to individual microwells. So we're having the wells again. Or antibody microarrays and allow for simultaneous detection of several cytokines from serum. So sample is serum, and we would want to know several, like if you would want to detect many cytokines, not just one, so we can use a that's why we have multiplex. It's multi, so it can have many cytokines detected in one setting. So current formulations allow for the detection of right now multiplex ELISAs allow 12 to 25 pro and anti-inflammatory cytokines detected in one reaction. And in the microarray format, each well on the slide contains a microarray of spotted antibodies with 4 spots for each of the 12 cytokines plus additional spots for positive and negative controls. The replicate spots allow for acquisition of reliable quantitative data from a single sample. But don't worry, you will not be counting the spots. There's an ELISA plate reader that will do all the counting for you. So we did the cytokine array test in during my master's, and we were able to try this one, which is really rewarding rather than just merely reading things from the book. We also have the new microbead assays. And new microbead assays permit the simultaneous detection of multiple cytokines, multiple meaning more up to 100 different cytokines in a single tube. Each bead type has its own, because it's using beads, and each bead type has its own fluorescent wavelength, which when combined with the fluorescent secondary antibody, we have the because we have the capture antibody and the secondary but biotinylated detection antibody, which is bound to a specific cytokine, allows for the detection of up to 100 different analytes in one setting. So I like this one really, if you want to have a lot of things in one setting. And the use of multiplex bead array enabled simultaneous measurement of representative pro-inflammatory cytokines such as IL-1, IL-1RA, 6, 8, TNF, Th1, Th2 distinguishing cytokines, interferons, IL-2, IL-4, IL-5, and the rest of those cytokines. So this one is really for someone if you would want to have a high throughput testing, then we can use new microbead assays. Don't worry if, oh, she haven't discussed the principles, the specifics, the procedures. No, because we will be having them in a more detailed format in our labeled immunoassays. So it's ELISpot is one of those assays that will be tackled or will be discussed. So this one is just presenting you that cytokines can be measured. Cytokines can be we can try to differentiate one from the other, and how and what are the ways in which we can do that is through those assays that I have just presented. So this is the end, and thank you for listening. Thank you for spending your time. I hope you have learned. Aside from what you have read, you have learned, and at first you might not be able to really understand things, but believe me, if you will read and view or listen to the lecture, then you can understand. If there are things that still boggle your mind or bother your mind, you can directly DM, or you can send a message to me. It's okay. And then thank you, and God bless everyone.